Hello, everyone. Today I will be talking about the Hendra virus, or equine morbillivirus, that's native to the western coastal portion of Australia. It's a member of the family Paramyxoviridae in the Hanapavirus genus. This single-stranded RNA virus are pleomorphic, meaning their structure is highly variable. The mechanism for infectivity is pretty interesting. The virus itself hosts upon a species of fruit bats called flying foxes, of which 47% of the fruit bats are carriers of the virus. Infected fruit bats may pass the virus in their urine and saliva. However, the exact mode of transmission is currently unclear. No human has ever been directly infected by contact with the bats. Rather, the intermediary of infected horses, hence the name equine morbillivirus, equine meaning affecting horses, is the cause of the infection to humans. According to the CDC, there have been only 7 cases since 1994 of human infection, and in every case, it has been those having extensive close contact with the infected horses. And all 7 cases have occurred in Australia, so this is not transcontinental or anything. It is very geographically isolated. In a study done to look at the outbreaks of the Hendra virus infection in horses up till 1999, several features were similar among the horses infected. One most prominent was, in all occasions, they were female thoroughbreds older than 8 years. As for its lethality and mode of infection, the Hendra virus case fatality rate in humans is 60%, which is incredibly high, and in horses, 75%. All individuals appear to have acquired their infection as a result of close contact with the body fluids of the infected horses. As an overview, this is the mechanism of infection of the Hendra virus. The fruit bat is the original host carrier and transmits the virus via urine or saliva to horses, and from there, horses in close contact with humans are the only currently known way of infection to the host, and that is usually through the bodily fluids. And as you can see here, there has not been a single recorded case of human-to-human infection. Okay, as for the Hendra virus itself, it's the single-stranded RNA virus, as we talked about, and the way it transmits itself is pretty interesting. So it has these G protein tetramers that are on the surface of the cell itself, and the way it gets access to the eukaryotic cell is through something called ephrin B2. Ephrin B2 is a highly conserved surface protein, meaning it's been selected for, or hasn't, I should say, not been selected against over time, and has been retained through the genetic makeup of the animal itself. We have them, horses have them, and fruit bats have them. So the way it does that, it attaches to the ephrin B2, has access, and begins replication. Here you can see in part A that the glycoprotein, the tetramer attached to the surface of the Hendra virus, is attached to the ephrin B2 of the eukaryotic cell, which is then bound and fused inside the membrane, releasing its single-stranded RNA, which is then replicated. And once replicated, it becomes part of the surface proteins of the eukaryotic cell, which then does something called budding. And budding is just, it enables viruses to exit the host cell and is mostly used by enveloped viruses, which the Hendra virus is, which must acquire a host-derived membrane enriched with the viral proteins that were transcribed to form their external envelope. Okay, I just wanted to address if it is in fact a viable option or an ideal bioterrorist weapon or a bioterrorist agent. So just to go through the list, its obtainability. You would have, first of all, it's geographically isolated to Australia. It's complicated to get out of the country. You would need to put it in a cryostasis state and, have that along with being able to get on a plane or a ship or something going unnoticed with the Hendra virus, which is highly unlikely, and obtaining a fruit bat and getting it across the border adds even more difficulty because you would need to, one, ensure it was infected with the virus, it's more than likely. 47% of the fruit bats are carriers of it. However, making it easy to store, no. Easy to deliver, not really, because so far it's only been known to be transmitted through horses to humans. Humans to humans, there hasn't been a single recorded case. There's currently no knowledge of it being able to be transmitted through an aerosol, making it not highly infectious. So the only two good reasons to even consider this as an ideal bioweapon is that it has an extremely high mortality rate, which is 60%, and induces fear. Possibly this would work in a less developed country where horses are used more extensively for transportation and, but in a more developed country like, say, North America, it's not very likely that it would cause as much fear and induce destruction or disruption as you would want. And there's much easier agents to obtain, better bacterial, viral options that would make it much more reasonable to use in an attack. And that is all. I wanted to thank you for your attention. I am now going to erase this before I startle somebody.